Hey guys, it's Dr. B. How you doing? Got a bit of a cold, so you might hear me sniffling around a little bit, but hopefully it won't bother you too much. This is just a review of Chapter 7 in terms of major aspects of the exam. Chapter 10 obviously is about cancer, so it would make sense to kind of give you a little bit of a background about exactly what it is. And you can look at an image like this, but basically I consider cancer 3 words: uncontrolled cell growth. It's very unlikely that we'll ever find a cure for, quote, cancer, because there are so many different types of cancers and so many different reasons why a cell would grow out of control. So there's no universal system that creates cancer cells. So that's just kind of one way to look at it. But if we start here more specifically in kind of what you're looking at here, it basically starts with some sort of mutated cell, which is right here. And it can develop into a benign tumor. And a tumor is a little bit of loss of normal cell division, but basically nothing really past that particular point. If they continue to lose other cellular controls in the cell cycle, they can become malignant tumor. And basically those are ones that invade tissues near them, because basically what's happening in the cell cycle is there's no checks and balances. So there's nothing to stop the cell from dividing, obviously after telophase. The cells aren't also normally organized, so they're not really functional cells. They're dysfunctional, and so if they happen to be cells that perform a task in an organ, you can see why that would be clearly an issue. So from that point they can move on and sometimes detach themselves to wherever they're located and metastasize, and that's where they can set up shop in other organ systems, and that's obviously not good. So there's just kind of a generalized progression of fundamentals of at least how a cell can become cancerous and lead to further stages of cancers. So how do they do it? Basically, I'm pulling some of this obviously from your textbook. I want you to concentrate on your textbook, but I'll add my own spin to it. You can see here you have a primary tumor at the top. And in order for a cell to become metastasized, it has to obviously dislodge itself from this primary tumor. And the way that they do that is sometimes cancer cells can reduce their ability to stick to neighboring cells. And so, normally, obviously, cells will stick to other cells. Look at you as a human. You're just literally a collection of 60 trillion cells all stuck to each other. So that's one of the major things, is they lose the ability to stick near each other. And once they do that, they can migrate, obviously to other areas, sometimes through bloodstream, sometimes through other lymph areas. So if a cancer cell is, swimming around the bloodstream, they can get trapped in areas such as your lung or your liver has very narrow capillaries, so a cancer cell might get stuck there and set up shop. And they can also express receptors that bind to sites that support metastasis, and that's obviously a big issue, especially in treating it. So a lot of these things will protect the cancer cells from your immune system. And that's one of the major reasons that they can avoid immune destruction. So once they stick, let's just say like stage F right here, basically what they can do is, you can see there's epithelial cells right here. They can retract or kill these epithelial cells to allow them to basically diapedese, which is a funny word, into the surrounding tissue area and to some secondary site. And then once they reach whatever that secondary site is, either by density or food, then they can set up shop somewhere else. And that's just a generalized overview of kind of how that works. You're going to want to spend a little bit of time on this slide, obviously knowing the difference of the omas, we like to call it. You can see here, an adenoma it would be a benign epithelial tumor. So these are just words you definitely want to, on your own check out. This is a pretty cool pick from your book, and just to kind of tell you, what this means. This is really talking about different regulators that can either increase or decrease cellular division. And you can see here anything with a green arrow is obviously going to increase the idea of cell division. Things in the red will decrease it. And so you can see here that like an apoptotic gene, such as you may have heard of like P53, it's a tumor suppressor protein. This would definitely be one that would stop cell growth. And there's actually a disease called Li-Fraumeni syndrome, which is where you have a mutated version of the P53 and you can't, your cells can't stop the process of becoming a tumor once that happens in some cases. BRCA1 and BRCA2 also can happen the same way. So this is just really kind of a neat overarching picture to show you that some, regulators that can increase proliferation and decrease. And you can assume that if some of these things were to go wrong, you can imagine what the outcome might be. So this slide I'm going to talk a little bit about telomerase. As you know, telomeres are the ends of chromosomes, and generally the older we get, our chromosomes get shorter and shorter after subsequent cell divisions. And so one of the fundamental ways you could just inherently statistically assume that cancer could be stopped would be because our chromosomes get shorter and shorter throughout our lifetime. And that's basically by the erosion of telomeres in our cells, and that can stop us from getting cancer because the cells basically run out of divisions. But clearly in the game of cancer cells, that's not necessarily the case. The cell can sometimes fuse together ends of chromosomes. It doesn't necessarily recognize the telomere end of the chromosome as being the end of the chromosome, and instead will treat it like a double-stranded DNA break and shove two chromosomes together, basically having two centromeres, which would be obviously further instability of the cell, but would avoid the idea of cell death. And obviously, inhibiting tumor suppressor proteins is yet another way as well. Oh, sorry about that. So basically, you can see here that proto-oncogenes and tumor suppressor genes are two classes of cancer genes, and proto-oncogenes are basically genes that are involved in some sort of normal growth or proliferation. So you can clearly understand that if something goes wrong with them, it could increase your chance of cancer. Tumor suppressor genes, actually, they're more examples of fighting against cancer. They code for proteins that slow down cell growth, which is kind of like the opposite of oncogenes in a sense. So when they notice things that go wrong, they halt the cell cycle. They cause other proteins to fix the error, typically in, a lot of times it will be in the S phase of interphase. But if something goes wrong with them, then they can't check the checks and balances of cell cycles, and therefore unchecked mutations would continue through the process of cell division. And so that's generally how both of those classes operate. Here's just an example of an oncogene activation that's caused by a translocation. So you can see here that you have chromosome 9 and chromosome 22. And sometimes there can be a translocation between these two regions of both these chromosomes, and you can see here that the ABL1 site has translocated over to the chromosome 22. We then call that the Philadelphia chromosome, or PH1, and this can cause chimeric genes and can be a cause of an oncogenic type of cancer. So this is a susceptible place where a translocation can affect an oncogene. So yeah, one other thing I want to kind of finish up this chapter with, and again, this is just an overview of the fundamentals. Obviously look at the exam study guide for more details. It did cover much of what's on the exam fundamentally. for chapter 10. This is basically a picture of what's called stratified medicine, and this is in all likelihood where, and it's already happened now, where the future of cancer treatment is going to be. What you would basically do would be to take not only the organ itself, the type of cell that the cancer is found in, but then also a genetic approach to identifying what particular mutation has caused a particular tumor. And then from that point, these tumors that will be divided into a major group, and these groups already have some degree of research that has predictable responses to certain types of drugs. So the idea is you might be taking a cocktail of different drugs that might target the particular tumor that you have. And you can see here this is just a particular example of the plethora of different methods or inhibitors, ways that we can treat the cancers based on what's wrong with the cells. So you see, if you have a tumor promoting inflammation, then you can use selective anti-inflammatory drugs if that's what that particular drug or tumor is showing. So that's just one little example of where medicine is headed and where a lot of the research is going. I want to end with this slide, which is pretty amazing. It's at the end of your chapter. This is an example of a patient that had therapy for a melanoma using what's called a BRAF inhibitor. And what this is, as you can see on the left, is this is glucose metabolism. And what they've done basically is illuminated, put a dye into the glucose, and using a tomography scan, they can see activity of basically where the radioactive glucose is, and that would be showing you cellular respiration at very high levels, basically. So you can see here on the left the cancer cells, and then after a treatment with this particular inhibitor, you can see that it was very effective in decreasing, at least within 2 weeks, the activity of some of the cancer cells that required that BRAF to function. So hopefully some of you will be our future scientists and help us find cures or treatments like this.